Yeah, good morning, everyone, and welcome to the crop hour for this week's wheat and small grains week. This is our second day. We started this week yesterday, um, and we'll have one more day tomorrow. And today we'll be discussing wheat diseases in the first half of the hour, and then second half of the hour will be the plant variety protection and seed certification and what all entails in doing that, and what are the things that restricts us from doing, and what are the things that we can and cannot do. And we have multiple speakers today. For the first half hour for discussing diseases, we have three speakers. Madelyn Shires, our plant pathology extension specialist, who joined SDSU Extension very recently. She's been here for just for a few months. And and two other speakers, Ashely Frederickson and Schneider Mahecha. So they will be, there is a video for Schneider's part, but Ashely and Madelyn will be talking on online. And before I give my hand this over to Madelyn, I would like to bring to your notice a couple things. If you look at your screen on the bottom side, there are a couple of, two things with a little comment box type icon, Q&A and chat. Feel free to put in any questions, comments on that, and after each presentation we'll go through that in order. And again, the second half of the presentation will be from Neil Foster about seed certification. So you can put in your comments and questions as we move along, and then we'll go and discuss that and address those at the end of each presentation. So now, without further ado, I'll um, let Madelyn take over from here.
All right, good morning, everyone. We are going to start off with Snyder's video recording. So give me just a second to get that screen shared, and then we will get started. All right. So here we go.
Hello everybody, my name is Mahecha. The name of my presentation is Efficacy of Essential Oils on the Management of Fusarium Head Blight in spring wheat. This study was part of my research as a SDSU graduate student, so I hope that you enjoy this one. So as a little introduction, spring wheat Triticum isothiums ranks third among the United States fair crops in planted acreage, production, and gross farm receipts after corn and soybean. Also, it is a good resource of vitamin B, and also wheat contribute about the 20% of the total dietary calories and proteins worldwide. So the concentration of production of spring wheat is mostly in the north side of the United States, in the states of Montana, North Dakota, Minnesota, and South Dakota, of course. So in this case, the host, the wheat, Triticum is affected by the causal agent, the pathogen Fusarium graminearum, that affect the heads and produce the disease Fusarium head blight. So Fusarium head blight results in grain yield loss, seed quality reductions, and also the accumulation of mycotoxins such as cerealeon and deoxynivalenol. Here are some of the typical symptoms that we can see: the head blight, the spicules, and on the right, this is an in vitro Petri plate, the normal colors and fungal shape, the fungus. So, but how is the management of this disease? The management of this disease can be by field rotation with an on-host crop, seed treatments, variety selection, variety of resistant or tolerant cultivars, and also discounting of the disease in the field to have the timing of fungicide applications. Okay. But, okay, the widespread use or the use of traditional pesticides has resulted in the development of pest resistance, pest resurgences, and new pest outbreaks, and hazard effects to the environment. So to mitigate that, so some alternatives are the biopesticides. The biopesticides are classified in the Environmental Protection Agency, like in these three ways. In this study, we are talking about the essential soils that are classified as biochemical pesticides. One example is the Mahecha ternifolia. That is a tree where we extract the tea tree that is used like a biofungicide. So, but what are the essential oils? The essential oils are extracted from plants, seeds, leaves, stem, flowers, fruits, and the main compounds are terpenes, terpenoids, and aromatic and aliphatic compounds. 3,000 essential oils have been identified, where about 300 have been used commercially in the agriculture. So, but why the interest of us for these products, for these alternative fungicides? Well, it's known that these products, these essential oils, in this case, tea tree, lemongrass, and like a simple citratus, have shown antifungal properties against a pool of pathogens that affect crops in the agriculture. Also, those appears right now. This is the FRAC code, the fungicide resistant active committee, as a new item in the table. For example, in this case, Mahecha ternifolia, and also appear the derivatives compounds like the sesquiterpenes or terpenoids, as biological with multiple modes of action. So that means that we have a new tool to use against for the control of fungal disease in field crops, in generally in crops. So there are approximately 40 products in the USA market with essential oils, and they're mainly compounds used for the control of plant disease. Two examples are, for example, Thymox with thyme oil and Thymorex with tea tree oil. So I'm going to try to summarize this. This is part of our experiment. We started first an experiment in vitro where we tested the minimal inhibitory concentration, the minimal fungicide concentration, the spore germination, and the volatile activity of several essential oils, compounds, and commercial products with essential oils. After to identify the minimum inhibitory concentration, the concentration that controlled the fungals in vitro, we increased that and made some experiments in the greenhouse. And finally, with those concentration tested in the greenhouse, we tested those in the field trial, and we got the variables that we took was the Fusarium head blight disease index, the yield, the FDK, and the deoxynivalenol analysis after the harvest. So some of the treatments that we used was tea tree, lemongrass, pure essential oils, two commercial products that I showed before, Thymorex and Thymox, and two compounds that are present in the essential oils, like carbachol and thymol. So here in the minimum inhibitory concentration, in the axis, x-axis, you can see the micellar growth inhibition, where the treatment showed the 100% of minimal inhibitory concentrate, the 100% of inhibition that was called for us the minimum inhibitory concentration. In this case, for example, the treatment lemongrass with the yellow, I think so you can see, or orange color, showed the minimum inhibitory concentration in 1,000 parts per million in this case. And so here, the treatments that showed the minimum inhibitory concentration clearly was lemongrass, Thymox, carbachol, and thymol, a compound that of Thymox and carbachol, another compound of Thymox. In the greenhouse, here we tested also, we had a disease index. We have for this one, three reps where each rep was a cone, and each cone has 3 spicules. And this experiment was repeated twice, where here we evaluated again the disease index of that of Fusarium head blight, and here, of course, Brosaru showed the best performance against the disease. But interesting for us, the treatments Carbacol, lemongrass, Thymox, and Timorex, and Timor showed significant lower difference versus the treatment control with water. A summary in our field study was in the location Volga, was in 2021. We used the susceptible variety select. We inoculated the plants with corn and also with the spray inoculation. And we used also an irrigation system with the sprinkles to increase at maximum the pressure of the disease. So here in the first columns are the treatments, the second columns, the yield. Here again, Prosaro showing the best performance. However, that wasn't significant difference versus our treatment Carbacol, Timol, and Thymox, and lemongrass. The disease index in the field was lower in the treatments Tea-tree, lemongrass, Thymox, Carbacol, and Timol compared with the treatment control with water. Also for our FDK, the fungal disease corner, we found that the treatments, mostly of the essential soils, showing significant lower difference amounts compared with the treatment. Here, for example, we found 32 after the harvest, and here just 14, 10 in this case, Thymox 8. For the deoxynivalenol concentration, we played this in ELISA test. For each one, we had 4 reps, and was repeated just 1 year. But here, you can see the treatments of Tea-tree, lemongrass, Thymox, and Timol showed a lower amounts was significant versus the treatment. Again, Prosaro showed the best performance in this study. So as a little conclusion, I'm sorry to take more time. So the minimal fungicide concentration was found at 800 parts per million for Carbacol and Thymox, and 1,000 parts per million for Timor. We saw a reduction of the disease in the greenhouse of from 55 to 61% with the treatment Thymox and Thymol. All the essential soils and compounds showed a significant reductions with a p-value of 0.05 in the FDK compared to the treatment control. Tea-tree, lemongrass, Thymox, and Timol compounds showed significant reductions in the deoxynivalenol concentration compared with the treatment control. So thank you very much for your time. Thank you, Maddie and Connie, for the invitation. And if you have questions, please let me know. I am open, and I would like to share more of my research with everybody. Thank you very much. Thank you, SDSU.
Okay, so hopefully everybody kind of got to see what Esneider spent a few years working on while he was finishing his PhD. He now works in Illinois in a diagnostic clinic, and we're very excited for him. Next, we're going to have Ashely talk about bacterial leaf streak. So Ashely, you want to go ahead and get started?
All right. So hi, I'm Ashely Frederickson. I'm a graduate research assistant here at South Dakota State University. And today I'm going to be talking a little bit about bacterial leaf streak of wheat. So bacterial leaf streak is an important disease. It was first observed on barley in 1917, on wheat in 1919, and on rye in 1924. It's been reported to reduce yields up to about 40%, but most reports are around 10%. The causal bacterium is Xanthomonas translucens pathovar undulosa, and it's primarily seed-borne. It's spread by splashing or wind-driven rain, and it enters the plant through wounds or natural leaf pores called the stomata. A little bit about the disease cycle. So like I said in the previous slide, the bacteria survives on or in wheat seeds. So when you plant those infected seeds, the bacteria multiplies and spreads. And then your beginning symptom will look like a longitudinal streak that becomes visible early in the growth stage, but BLS, bacterial leaf streak, can also infect the plant at a later growth stage, mostly during grain fill. So some symptoms of bacterial leaf streak, that longitudinal streak that you begin seeing early in development is referred to as water soaking. It's an irregular translucent water soaked streak. And if you hold it up to the light, that lesion looks translucent. It will become chlorotic and eventually necrotic. Another symptom is bacterial ooze. In warm and humid conditions, milky yellow sap kind of oozes out of the lesion, and it will dry on the leaf surface, giving the streak a shiny appearance. Bacterial leaf streak is also commonly referred to as black chaff, and that's when it infects the glumes during grain fill, which causes a dark purple to black streak. And in severe black chaff cases, it may result in discolored kernels. A little bit about the disease management, because there isn't much for the management of bacterial leaf streak. It can be a pretty hard disease to control. Cultural practices, meaning rotation away from a non-host crop, is a good option for ridding your field of BLS infection. Straw from previously infected crops or a weed host can serve as a source of overwintering inoculum for field infestation. So by reducing the straw or incorporating it into the soil is a way to rid your field of that. The pathogen is short-lived in the field, so excluding that weed host and rotating to a non-host crop will contribute to reductions of inoculum in the field. Certified seed, so making sure you're using clean and pathogen-free seed is very important, since bacterial leaf streak, the pathogen is seed-borne. So the clean seed can help prevent infections and spread. Variety resistance, there is no 100% resistant variety to bacterial leaf streak. There are some cultivars that are less susceptible to the disease and like moderately resistant, but there is no 100% resistance. And then chemical control, there are no bactericides or cultural chemical controls for managing bacterial leaf streak, which is where my research kind of comes into play. So my research here at South Dakota State University entails screening antagonistic bacteria for biological control of Xanthomonas translucens pathovar undulosa, denoted as XTU, causing bacterial leaf streak of wheat. And so biological control is using another living organism for the management of a disease. So in my case, I'm using soil bacteria or bacteria found in soil to manage bacterial leaf streak in wheat. So I've taken 20 soil samples from South Dakota and 20 soil samples from North Dakota from previously infected with BLS wheat fields. And I've screened them and found bacterial isolates to test against the Xanthomonas translucens pathovar undulosa. So what you see here, the picture of the Petri dish, is a lot of my antagonistic screening. So I take the XTU and I swab it on the base of the Petri dish, and then I put plugs of my potential antagonistic bacteria that I've isolated from those soil samples. And then I look for that ring that's around the agar core. And that's a zone of inhibition. So that's stopping the growth of the XTU in vitro. In the next couple months, I hope to screen all these bacterial isolates against Xanthomonas translucens pathovar undulosa in vivo in a greenhouse setting. And then hopefully in the coming year, we'll get those to the field. So, yeah. Thank you. If you have any questions, my email's there, but yeah.
All right, so thanks, Ashely. I now have a few more wheat diseases to talk about for the next few minutes, and then we will have the diseases finished up. Okay. All right. So if you've watched Crop Hour the last couple of weeks, you have probably seen that I have been just trying to talk about some of the most frequently reported diseases in the North Central region. Today, we're just going to kind of jump into these. These are the top 5 reported wheat diseases for the Northern United States. Stripe rust is the most reported. It's the most frequently reported loss for wheat, followed by Fusarium head blight, which is what Esneider was talking about previously. Also, wheat viruses such as high plains disease, triticale mosaic, and wheat streak mosaic, tan spot, and Fusarium root, crown, and foot rot. So hopping into stripe rust, this is a fungal disease that pops up a lot of years. It's the name of the fungus is Puccinia, and I'm not going to attempt to pronounce that species name. The symptoms start as yellow flecks on the leaves, and as the disease worsens, the pustules develop into linear stripe-like patterns and develop into these yellow-orange spots like you can see here on the sample. Later in the season, the spores will turn black and they actually overwinter on that wheat material. So if you have stripe rust one year and leave wheat stubble in the field, there's a chance it will pop up again the next year. And this is basically what I was just explaining, that it can overwinter in fields, start from those spores, develop an infection, and then this kind of repeats throughout the year of developing spores and infecting more plants. All right, so that's kind of that disease. Moving into a couple of wheat viruses, because we do see those pop up from time to time here in South Dakota. Barley yellow dwarf, it's a disease that's caused by multiple viruses in the Luteoviridae genus. The symptoms are red, yellow, or purple discoloration appearing on leaves, especially the flag leaf, and you can see that right here in this picture. You may also see tip burn, distorted leaves, and underdeveloped root systems. The symptoms are most likely to appear on cool days with high light intensity. So as we're starting up our spring season here in South Dakota, these viruses are spread by aphid feeding. The management for these are to use resistant varieties and manage other hosts such as weeds in your field. And then here's a few more examples, and you can kind of see that reddening and yellowing on the tips of the leaves and on the flag leaves. So it can be a problem. We don't see it quite as often here in South Dakota, but you never know what the weather is going to do this year. Some other wheat viruses, there's the mosaic virus complex that's caused by those 3 different viruses, and they all cause similar symptoms. This is an example here of wheat streak mosaic virus. So we're going to briefly cover these. So the biggest problem with these viruses is that they like to infect, multiple viruses like to infect the same plant. And that, what we call a co-infection, can be devastating to yield if there's 2 or more of these viruses. There can be losses in spring and winter wheat if the virus levels are high. And it's kind of hard sometimes to pin down the symptoms because they're very dependent upon the variety and which viruses are co-infecting in the plant, what the temperature is like. So it can be kind of hard to figure out exactly what's going on. So here's some symptoms of the different viruses. So wheat streak mosaic virus, which is the one that a lot of us have probably heard about because it does show up here in South Dakota pretty frequently. Stunting and streaky discoloration on the leaves. It progresses more with warmer temperatures. Symptoms of high plain disease, those are small spots that turn into mosaic patterns, and then it turns into necrosis or dead tissue. And then triticum mosaic, it's similar symptoms to wheat streak. The heads may be sterile or not form if the virus infection is really bad. And it can also lead to severe stunting and death. Tan spot is another fungal disease that does pop up here in South Dakota if the weather conditions are good. The symptoms of it starts as small brown spots that develop into tan necrotic spots with a yellow halo. So you'll have tan dead looking tissue that has a yellow halo on it. And you can see that kind of happening here on these, especially that. The development of tan spot is favored by wet, windy weather, and tan spot can pop up at any point in time during the season. Here's some more examples of tan spot. As you can see really, really well, these are some of the leaf symptoms you'll see. These are actually the fruiting structures that will develop on the stems of the wheat and produce more tan spot spores, and so more inoculum in the field to cause more disease. Moving into some fusarium rots, excuse me. This is caused by members of Fusarium genus, so it's similar, it's the same genus as Fusarium head blight. Seedling blight can occur if Fusarium is present on the seed, and it does sometimes get carried on the seed. Seedlings that get infected tend to be reddish and have poor vigor or tillering. In older plants that get infected, a dark brown to red lesion that will be apparent near the first node of the plant, so kind of in that first few inches above the soil line. Humid conditions drive the symptoms, and stems can become spongy and decayed with a pink fungal growth on the stem. And I've got some pictures next. So you can see here that discoloration that's happening around the first node. And this is happening because the fungus is preventing the movement of nutrients. And this is some other examples. Another one that we do see most years is this disease here, Stagnospora. It causes both leaf symptoms and gloom symptoms. So leaf symptoms start as small yellow spots that become grayish brown colored and elliptical shaped. The lesions will have a chlorotic, so a yellow margin, and you can actually see some brown fruiting structures that start to develop similar to tan spot. Again, this is the gloom symptoms. So the symptoms start as purple to brown discolorations, and the glooms appear dirty or water soaked. And it will develop fruiting structures to produce more spores on those glooms. Symptoms of this disease can be seen from tillering to ripening, and the seeds can also have symptoms, and they're typically discolored. We don't have a ton of time to go through this, but this is a good example of how this disease continues in the field. Rain and wind is kind of what helps spread the spores and make the infection worse. The fungus does survive over winter in the wheat residue and on infested seed lots. This is another fungal disease, Septoria triticide blotch of wheat. The symptoms start as tan to brown lesions with yellowish halos, similar to many of our other spot diseases. The lesions, again, will become necrotic, and you will see some of those fruiting structures, those little black fruiting structures popping up on the dead tissue. When it's humid, the fruiting bodies may ooze, and this disease is most likely to occur with cool, wet weather. Here's some examples of it. So you can see the spots are shaped just a little bit differently. There's those fruiting structures that are starting to develop in the spots. The final one that I'm going to mention is powdery mildew. It does pop up every now and then, especially if we have wet weather. The symptoms are fluffy white to gray growth on the tops of leaves. Yellow leaves may be seen at the bottom of the plant. If the infection occurs very early in the season, it can affect yield, and it's most commonly occurs on winter wheat. So just a final slide for a plug about the plant diagnostic clinic. If you don't know what you're dealing with, it can make management decisions very challenging. So don't guess on pathogen problems, send samples in for testing. I am also the director of the plant diagnostic clinic here on campus. So if you have any questions, need to send in samples, feel free to reach out and contact me. The resources used in this talk all came from the Crop Protection Network website. And there's my contact info. And Connie Shrunk is our plant pathology field specialist stationed in Sioux Falls. So if you need any information or anything, feel free to reach out to either of us. And yeah, I think with that, we are done.